Assalamualaikum and very good day. Hi everyone. My name is Nurdamia Izani binti Reseei. I would like to present our research and the title is Biodegradation of Methylene blue dye using Trichoderma Reseei SP. So for the next slide, you can see there is a project outline which is the content that I will be presenting to you guys. The first is the introduction which consists of methylene blue dye, Trichoderma Reseei SP, the problem statement, objectives and scope of research and benefit to user and society. And then I will be presenting about the methodology, results and discussion, conclusion, and lastly, the award and recognition received. So for the introduction of methylene blue, it is maybe used for biological and chemical purposes such as treatment of methemoglobinemia. So methemoglobinemia is a blood disorder in which too little oxygen is delivered to your cells. And then it is also used to treat cyanide poisoning, urinary tract infection, and made as biological dye and stents. So, one of its properties is it is able to penetrate cell tissues for medical identifications. However, we have negative effect because the overdose above 5 mg per kilograms of methylene blue will cause serotonin toxicity. So, serotonin is a type of hormone inside your body. It will cause cardiovascular problems, skin staining, nausea, and also anemia. So the next slide is about the Trichoderma Reseei SP. It is a type of fungi and it is one of the most prevalent culturable fungi which is also present in all soils. The decolorization of methylene blue is permanent in pH 8 and at temperature of 27 degrees Celsius. All of the fungi sample that we collected before, we found that fungi Trichoderma Reseei SP is the most prominent fungi to biodegrade methylene blue dye. So the next is the problem statement. Chemical dyes are a major contributor to current pollution. By 2018, 38% of water sources are likely to be contaminated due to chemical dyes. Demand for water treatment technology is high. Various conventional methods such as physical, chemical and biological processes have been tried for the removal of dyes from aquatic media. Methylene blue dye is also broken down into smaller particles in water sources. Water is also polluted for domestic use and living organisms, which means that we cannot use the water more efficiently, and other water treatments need high maintenance. So, the objectives of our research is to investigate the effects of physicochemical parameters on decolorization of methylene blue dye, such as the carbon source concentration, which we use in this research are glucose, and we also focus on the nitrogen source concentration, which the yeast extract. And then to analyze the methylene blue dye decolorization by Trichoderma Reseei SP using UV spectrophotometry and record the data. Our scope of research is Trichoderma Reseei SP is the fungi chosen as one of the prominent fungi for methylene blue decolorization. Methylene blue dye is the main focus in this research for decolorization process. The physicochemical parameters that are measured are the concentration of carbon source and also nitrogen source. So the next is benefit to user and society. Biodegradation of methylene blue ensures sustainability of ecosystem, thus preserve our environment for future generations. This research increases public awareness on the importance of protecting our environment for greater life. So we will move on to the methodology. The first thing that we did is culture fungi in agar solid medium. So Trichoderma Reseei SP fungi is grown in agar medium. In the agar medium, the materials used are nutrient agar for 20 grams per liter, glucose 20 grams per liter, chloramphenicol 300 milligrams per liter, methylene blue dye 50 grams per liter. Chloramphenicol is antibacterial, so it can prevent contamination of the culture medium from bacteria. After that, the samples are replicated for 5 different samples and the fungal samples are kept in incubator for 27 degrees Celsius until it is fully grown. And we can move on to the next methodology, which is preparing the liquid medium. So liquid medium is prepared from seed culture and petri dish culture. 2 grams of malt extract powder is added in making of seed culture in 27 degrees Celsius. the optimum temperature. The seed culture is a fungus that has been taken from agar medium. We use scott borer to punch the seed culture and insert 3 seed culture plugs in liquid medium. The materials are then prepared from 20 grams per liter of glucose, 20 grams per liter of yeast extract, and for 120 rpm. So for the effects of physicochemical parameters, which is the concentration of nitrogen source and carbon source, so we manipulate these two substances throughout the experiment to identify the optimum value. Optimum concentration of carbon source is important as it acts as co-substrate in enhancing and elevating biodegradation of methylene blue, and optimal nitrogen source is to provide nutrients for fungal growth. Excellent fungal growth will increase production of enzymes that enhance biodegradation of methylene blue. Initial dye concentration, pH value of 8, temperature at 27 degrees Celsius are kept constant throughout the experiment. For the carbon source concentration, we manipulate into 5 mg per liter, 10 mg per liter, 15 mg per liter, and so on. The same goes with nitrogen source. We kept the temperature constant at the 27 degrees Celsius, so the carbon source is glucose and nitrogen source is yeast extract. So, for the collecting of data, the effect of physicochemical parameters is analyzed using UV spectrophotometry, and the data obtained is collected and we analyze it into the graph that you guys will see, then the results and discussion. So here is the results. As you can see from the graph, the percentage of methylene blue decolorization and its concentration of carbon source milligrams per liter. The rate of the biodegradation of methylene blue is highest at concentration of 15 mg. Therefore, 15 mg per liter of glucose is found as the most optimum concentration for the biodegradation methylene blue dye. So the next slide is about the nitrogen source concentration. We choose 10, 20, 30, and 40 because the percentage of methylene blue dye decolorization increases as the concentration increases. So we choose the most optimum, which is 40 mg per liter, and combining these two results, the carbon source and nitrogen source, we can actually create a very ideal fungi for decolorization and biodegradation of methylene blue dye. So lastly, the conclusion, Trichoderma Reseei sp showed the best performance in decolorizing methylene blue dye with carbon source concentrations of 15 mg and 15 mg per liter and nitrogen source concentration of 40 mg per liter. And this is the awards and recognition of our project. So our project has been presented at INSAN Junior Researcher International Conference and Science and Technology Conference in 2020. So that's all from me. Thank you so much.